Hey there, super nerds. Today we're going to talk about Mendel and alleles. So, what's the point? Basically, by the end of this, you should be able to outline the work of Gregor Mendel and what his discovery of dominant and recessive traits. You want to be able to define genetics and heredity, and you also want to be able to describe the effects of the environment a little bit on these, and you should be able to predict the outcomes of simple inheritance using Punnett squares. So, genes, basically, they're the unit of heredity. Okay, we know this already. You know that genotypes are the genetic combinations of individual, and you also know that phenotypes, they're the physical expression. So genes combined with the effects from the environment. All right, so let's go through some simple definitions. So an allele, basically these are different versions of a gene. Diploid cells have two alleles for each gene. They can be the same or they can be different. So here we've got two separate chromosomes. It's a pair of chromosomes. And we've got allele A and allele B. And you'll notice that they're in the same location. If you're homozygous for a particular gene, this means both copies are the same. They are identical. So you've got the same allele. So here we've got allele for phenotype A, allele for phenotype B. That is not homozygous. If you are heterozygous, this means both copies of that gene are different. So you've got different alleles. This guy here is heterozygous because it's allele for phenotype A, phenotype B. And we can see how this is inherited just here. So if we've got a dominant allele, this means if you are heterozygous, so you have two separate copies, then the dominant allele will be the one which is expressed. Okay, if you're homozygous for the dominant allele, it's obviously the one that's expressed because it's the only one you've got. But if you are heterozygous, the dominant one is expressed. If you have a recessive allele, this means that it will only be expressed in a homozygous individual. So, for example, here we've got, this is the allele for blue eyes, which is recessive, brown eyes, which is dominant. Okay, so if you have, this is a heterozygous person, brown eyes, blue eyes allele, they will have brown eyes. If you are homozygous for brown eyes, you will also have brown eyes. But if you are homozygous for blue eyes, that's the only time you will have blue eyes. It is much more complex than this in real life, but this is how we explain it for the moment. This is the example we like to use. All right. So next we'll talk about what a dihybrid is. A dihybrid cross. So hybrid cross is when we go this one cross with this one. All right. So two separate alleles. A dihybrid cross is when we're looking at two separate genes. So two separate traits at the same time. That'll be important in a little bit. All right. We do it with a lowercase and uppercase letters. So very quickly, this would be brown eyes, that's dominant, so that would be capital B, and this would be lowercase b. So the genotype for this one would be, we always do the dominant one first, it doesn't matter the order. This one here is brown eyes, so it's B. That's how you write the genotype for it. And this one is done like this. And this is important to know. All right. Now, here is the example with the P's that we're using. So we've got purple flowers, white flowers, and this is a separate chromosome. So you see tall stem or short stem. See, that was the dominant one. That one's the dominant one. In a dihybrid cross, you would look at both of those at the same time. Now, Mendel actually looked at 6 traits. Okay, so he looked at flower color. He was a monk, by the way. We'll talk more about him in class. He was a monk from the 1800s. He looked at flower color, flower position. Is it on the axial, so over here, or is it terminal? Is it on the end of the stem? Seed color, seed shape, round or wrinkly. Pod shape, inflated or constricted. Pod color, green or yellow, or were they tall or short or dwarf? And what he found was if he crossed one purple with one white, so a pure breeding purple, pure breeding white, which means no matter how many times you cross them against each other, you always get the same colors, he always got purple. Axial and tall and terminal, he always got axial flowers. Yellow and green, he always got yellow. Round and wrinkled, always got round, and so forth along the way. So let's look at what he actually did. These were his experiments, and you need to have this down. Okay, so here's his parental generation. You need to have all of these. So you've got true breeding, so we know its genotype is TT, lowercase t, lowercase t. So this cross with this always gave us these guys here. Okay, so we then make those siblings. We put those siblings together in our F1 cross. So here's our F1 generation. We're now doing an F1 cross. So F1, it means the F is for familial, which we don't need to know, just know it's called the F1. So we put that with that, and we start to get a mix. Okay, so we've got a tall because it's dominant. All right, so we've got 1 out of 4 of these is homozygous for the dominant, 1 out of 4 is homozygous for the recessive, 2 are heterozygous. What this actually means for the phenotypes, though, is that we have 3 tall, 1 dwarf. So the genotype ratio is 1 to 1, homozygous, heterozygous, homozygous. But the phenotype ratio is 3 to 1. And that is always the case. And if we look at his results, we'll just get rid of all the ink. If we look at his results, what actually happened was he got roughly the right ratio of 3 to 1 every single time. What we actually find with Mendel is that his results are perfect. They're a little too perfect. So we think in the end he might have cheated, and that's not very cool. But when we've redone his experiments later on, we still get roughly the same results. It's just not quite as perfect as he did. So we can predict them. Okay, we can predict them using these things called Punnett squares. So here we have the purple flowers. Okay, so we're giving them B and lowercase b. Because we have what we call we're using the heterozygous ones here, so it's actually BB cross with BB. We know that each offspring gets 1 gene from each parent. So from the mum, sorry, it's the dad, sorry. From the father, it could get either this one or this one. From the mother, it could get either this one or this one. So we're predicting the likelihood, the chances of certain offspring. Okay, so you've got a 1 in 4 chance of getting a homozygous purple. You have a 2 in 4 chance, or a 1 in 2 chance, of getting a heterozygous purple. And you've got a 1 in 4 chance of getting a homozygous white. Over here, if we're going tall and short, it's the same thing. So the genotypic ratios are 1 to 2 to 1, and the phenotype, so the physical expression, is 3 to 1. Now, we can do this with multiple traits, which just means our Punnett squares, this is a Punnett square, our Punnett squares get a bit bigger. So here's the father, here's the mother. From the father, which is, the fathers are all, and mothers are both this, so F1. With the mothers, with the father, we have 4 possibilities. We can get an AB, capital A, capital B, capital A, lowercase b, lowercase a, capital B, lowercase, lowercase. And here we can break down our results. So with this one here, so there's 4 by 4, so it's 16. So we have a 1 in 16 chance of this guy. This one though, which is dominant homozygous for the A's, heterozygous for the B's, we can go 1, 2, and we've got a 2 in 16 chance. Okay, and we can see as we go along, we get more and more, so a pattern starts to emerge. All right, so that's what we've got. That is our basic intro to how to do these. We're going to practice these in class. If these seem confusing now, don't stress, we will practice these in class.